In this part, we are going to study about our artificial vegetative propagation. In our previous class, we already know about our natural vegetative propagation, and we also gained some knowledge about vegetative propagation in natural methods. We also know that the plant parts, just like your stem, your root, your some of the examples of bulb, some of the rhizomes, and some other parts like your offset and some other parts are also helped in your vegetative propagation in your natural method. So from these techniques are related, but in artificial method these techniques are used in a man-made process. In artificial methods, a number of hormones, a number of nutrient mediums, a number of techniques are used by we human beings to develop our plants in our gardens, in our natural processes, in our fields also. So this is otherwise known as your artificial method of vegetative propagation. As we studied that this artificial method of propagation also used a number of subheadings stuff like your cutting, Gootee, your grafting process, layering process, and lastly your micropropagation process are also used, which are otherwise known as your tissue culture process. So in previous class we already know that stem is mainly helped in your vegetative propagation. Some other parts are also used, but in firstly we know that stem is helped in vegetative propagation. So after knowing that technique, we artificially used that technique in your natural plants also. So our first part is cutting. In cutting, in process of cutting, artificially we cut a healthy stem part of a plant, and this cut should be in slender in nature. That means the cut takes place below the node part. Here the diagram also present here. The cut takes place below the node part, and the cut is in slender in nature. That means in downward cut takes place as a plant from the part also, from the node part also. As we know that node is a sensitive part of the plant, and it also helps in growing of adventitious roots. So we don't want to damage that part also. So we use this types of technique in healthy parts of stem in cutting nature. After cutting the stem, we dipped it in a auxin solution, as auxin as a growth hormone. It helps the plant to grow number of roots, and after some days we saw that the node part and the slender cutting part also developed a number of root systems also, and developed a new plant itself. So in cutting, these types of techniques are helped in your artificial method, in your gardening cultures also. Nextly, in your layering part. Layering is mostly divided into two types. One is your layering of ground, that means otherwise your ground layering. Another one is your air layering. In ground air layering process, we used mainly some types of flowering plants, because flowering plants are mostly fleshy in nature. They are not rigid. In this technique we used some types of flower plants, because if we take an example of your jasmine, jasmine plant is mostly fleshy in nature and not that much rigid. So in this process we bend the branch part of this jasmine plant, and the precaution is that the whole plant part is not used in a layering process. We just bend it, and it is important that the node part is deep in soil. That means we covered some amount of soil in it, and after some days we saw that from the node part there is a number of roots arise, because we know that from the node part adventitious roots are developed. In your stem part also adventitious roots are developed. In your sugarcane you also saw that a number of adventitious roots are developed from the node part. Just like that, here also the number of roots are developed from your node part. And after some days there is a teeny plant is developed, teeny jasmine plant is developed, and the parent plant is itself from their cut from the new parts, and there is two different plants are raised. So this technique is used mostly in flowering plants. Nextly, we study about our air layering. Air layering is takes place mainly in your rigid plants, such like your mango, and some types of some fruit plants like your guava, orange, and some lemon, such like your citrus plants are also some of the examples of your artificial layering. In artificial layering process, in air layering process, artificially we used a simple healthy branch and use some cut down some of the dry parts of this. That means your bark part is removed out from the plant. Here we take a mango plant. Say this is an example of your mango plant. If we take a simple healthy branch, then we cut some types of some barks from the healthy part. That means in this part, some part is removed out, some bark part is removed out. That means these parts are quite dry in nature, so this part is removed out, and to fulfill that gate, we used some types of nutrient medium containing substances naturally, that are your cow dung, some mosses, and some nutrient mediums are also used. And from after filling the gate, we cover it tightly with a number of cottons and wrapped it. And this wrapping process is authorized. The wrapping part is authorized known as a Gootee. After some days, we saw that there is a number of roots are arising from that part, and after fully developing that part, we cut the branch, and the parent and the new offspring plant which are developed are cutting down from your parent plant and develop a independent plant also. This is authorized known as your air layering process, authorized known as your Gootee also. So in this part we are already covering the cutting part. Next is our layering part, which is your ground layering. Another is your Gootee part. Nextly, your grafting process. Grafting means the process in which we are used two different characters, two different desirable characters to develop artificially in a single plant. If we take an example of your in flowering plants, if we take an example of a simple rose which are developed in natural process, a simple rose, and which are not which is a wide variety. That means its root system and shoot system are quite good, and developmental structure is quite good, but its flowering parts are not such much good. There is no attractive parts, there is no flowering attractive whorls are developed, there is no attractive flowers are also there, and flowers are not such good. But a teeny plant is also developed in natural process, another plant which is not good in root system and not good in shoot system, but its flower is very good in nature, and its flower is quite healthy, and the bracts are aggressively colored, and the bracts are quite attractive in nature. If we want to combine these two types of character, if we combine these two type of character in a single plant, we use this type of technique which is authorized known as your grafting technique. Here we combine two different character in a single plant, and it is also one type of cutting and layering process, just like that. And here we just use not going to use a single plant, we use two different combined with desirable characters also. Nextly, we are going to study about our micropropagation. Micropropagation is authorizedly it is quite similar to your tissue culture process, because its definition shows that it is a rapid vegetative propagation which is developed from the tissue culture process only. If we take and if we give some examples and saw that it is a technique, it is a quite easy technique and quite rapid technique also. A number of nutrient medium, a number of classification structures, a number of developmental structures are also used in tissue culture process because it is a vast process. In this process, we saw that if we take a plant part, say your egg plant, because in this process, those parts which we mostly use the growing plant parts which are quite healthy. We used quite healthy plant and quite growing healthy plants as our micropropagation process, as your tissue culture process, because in this process there is a developmental structure takes place of the cells, and we also know that the cells are divisible in nature. If we give some types of nutrient medium, some type of nutrient medium to the healthy plants and some types of healthy cells also. This is a quite long process, and it is rapidly divided itself, and it gives a natural developmental structure of a plant. So we take such type of plant parts which otherwise known as your egg plant. From the egg plant we take some types of cells, and the cells are dipped in a nutrient medium which is otherwise known as your culture medium process. This is a simple practice. We dip it down, and after some days we know that we are going to see that the cells are divisible in nature. So cells are divided into multiple times, and gives a thallus-like structure which is otherwise known as your callus, and the nutrient medium is otherwise known as your callus, and the plant which are used in this technique otherwise known as your egg plant. So the plant part is divided to form a callus culture which is known as undifferentiated mass of tissues. That means there is a mass of tissues that takes place, which is otherwise known as a thallus. Then the the callus is developed to a plantlet. That means the cutting part of this plant. That means the growing. After growing some days, the cells are divided, and a mass of cells, a chunk of cells takes place in a single place. After removing that chunk of plants, chunk of cells, we just cut down in a petri, and the small pieces are going to dipped in a nutrient medium, such like your growing nutrient medium in the laboratory in test tubes, and these parts are used to develop new plants, new baby plants also. So this, after some days, these plantlets, it is otherwise known as your plantlets. The small plant parts which are developed from this callus culture is otherwise known as your plantlet, and the plantlet are going to develop your free plant, which is develop your new plant also. That means we simply cut down a simple. In this process, we use a simple growing tissue, and growing tissue is dipped in a solution which is containing a number of nutrient mediums. After some time, it causes a callus-like structure, and the callus is again cut down to a number of plantlets, and the plantlets are dipped in some types of nutrient medium to give rise to a number of new plants, and the free plants also. We also say that free plants. So it is a simple process, and micropropagation process are mostly used in laboratories to develop a number of new plants from a single cell, to grow a mass of cells and grow a number of new plants, new baby plants. So it is quite differentiated process, and quite useful to develop a plant mass also. So in this topic, we particularly know various types of artificial methods such as your cutting, layering, grafting, Gootee, and nextly your micropropagation also. These are some of the examples and some of the techniques which are used to develop the new plant from these types of artificial methods, and this is all about your artificial vegetative propagation. So this is all about your video. Thank you.